Hello everyone, welcome to MMBS-111, Introductory Microbiology. This is mini lecture 10.3. We're going to be talking about antimicrobial resistance. There are 4 learning outcomes for this mini lecture. The first is discuss 2 possible ways that microbes acquire antimicrobial resistance. The second is to name 5 cellular or structural mechanisms that microbes use to resist antimicrobials. The third is discuss at least 2 novel antimicrobial therapies currently under investigation. And finally, discuss some ways to keep normal flora healthy during antibiotic use. So, drug resistance can be acquired in a number of ways. Essentially, microorganisms begin to tolerate a certain amount of drug that would normally inhibit its growth or kill the organisms. And this happens because microorganisms are very versatile and adaptable. They reproduce very rapidly, and that means that mutations tend to happen more frequently. And as we discussed in Chapter 8, mutations tend to normally be harmful, but not always. They can also be beneficial or cause no effect. And in the case of drug resistance, sometimes those mutations allow the organism to resist a certain antimicrobial. The ability to resist a certain antibiotic can also be intrinsic, so it can naturally occur in that organism without ever being exposed to the antibiotic. But it can also be acquired from other organisms or through genetic mutations. Drug resistance was identified fairly early on after antimicrobials began to be used. So after the initial use of penicillin, doctors began to see penicillin resistance fairly early in the 40s during the treatment of wound infections during World War II. In the 80s and 90s, it became even more prevalent and on a much larger scale. So more organisms were demonstrating drug resistance and to a much greater variety of drugs. Microbes become newly resistant to a drug after one of the following activities occurs. Either the organism experiences a spontaneous mutation while replicating its chromosome, or it acquires the drug resistance gene through horizontal gene transfer from other organisms. So let's look at these 2 methods for developing drug resistance. In chromosomal drug resistance, we're talking about a spontaneous random mutation that occurred that happened to be beneficial for the organism, allowing it to resist a particular drug. Sometimes this results in slight changes in drug sensitivity, so it might be sensitive, it might be resistant, to a drug at a low dose, but still sensitive to the drug at a higher dose. There are also organisms that are known as persisters, and these organisms are typically found within biofilms, but they could be planktonic. And these organisms have developed the unique ability to slow or stop their metabolism in the presence of the antibiotic so that they they're not harmed by that antimicrobial agent. They're able to survive. So once the antimicrobial has left their environment, then their metabolism picks up again. There are also organisms who develop drug resistance through horizontal gene transfer. They gain that gene from another organism. And these genes are typically found on plasmids. They're called resistance or R factors. And they can be transferred to other organisms through conjugation, transformation, or transduction. sometimes an organism will naturally have this R factor encoded on a plasmid. Because we derive our antibiotics from other microorganisms who are naturally found in the environment, these organisms are likely exposed to that antimicrobial before we've ever used it as a therapy for treating an infection. So they may have that R factor naturally. Transposons can also produce resistance because they duplicate and insert genes in the chromosome and into plasmids. So they could insert a drug resistance gene into a plasmid that could then be transferred to other organisms. This type of development of drug resistance is the method that leads to the greatest and the fastest spread of antibiotic resistance through different species of organisms. This is a video that I've inserted here. I'm not going to show it as part of the lecture, but I would encourage you to watch it with the PowerPoint. This video was created by Harvard Medical School, and they were investigating how organisms evolve drug resistance and how quickly they are able to acquire resistance, not only to a particular drug, but to differing concentrations of that drug. So I would encourage you to watch this. It's pretty amazing how this plate is constructed and how rapidly these E. coli are able to spread across a plate containing antimicrobials at concentrations that should be lethal. So this diagram is from your text, and it illustrates the number of ways that microorganisms have developed mechanisms to resist antimicrobial therapies. The text in red I added as a summary really of what these mechanisms do. So the first way is that the organism develops a new enzyme that is able to inactivate the antimicrobial. And this is what beta-lactamases do and penicillinases. They inactivate those particular antibiotics so that they're no longer effective. Another way that bacteria are able to develop drug resistance is that they have altered the surface receptors that naturally recognize that drug and bring it into the cell. So the receptor has been changed or altered in a way that it no longer is able to recognize that particular chemical, so it doesn't enter the cell. Some microorganisms are able to produce a pump. These are called efflux pumps, and they actively pump the antibiotic out of the cell. So the receptor is still present and the antibiotic is still entering the cell, but this organism has developed a pump that immediately pumps that antibiotic out of the cell. And you generally find this mechanism in organisms that are multi-drug resistant. So they might have multiple pumps that just pump each antibiotic back out of the cell. Some microorganisms have changed the binding sites on enzymes or other structures like ribosomes so that the target drug that typically recognizes that binding site, it cannot bind any longer because the site has been changed. And the last mechanism is that the organism has changed its metabolic pathway. So it no longer follows the same pathway for developing a particular product, whether it's folic acid or a protein. It has changed the pathway and uses different intermediates in order to produce the same necessary product. So the drug now is no longer effective because it doesn't recognize the new intermediates in that pathway. Drug resistance has a lot to do with evolution and natural selection. If you look at the image here, you can see that we have a plate of organisms with a number of colonies. All of the white ones are not drug resistant, but the two red ones are drug resistant. And this is typical for many populations of microorganisms. There will be colonies or individual organisms within that population that are going to possess naturally resistance to a particular antimicrobial. When we use that antimicrobial against that population, the ones that are not resistant are going to die. And now we have left ourselves with the organisms that are resistant. So those organisms are going to reproduce, and now every single member of the new population is resistant to that drug. And it happens very quickly. So this problem is a worldwide problem, and it has a lot to do with how antimicrobials were managed in the past and how they are currently being used. It has a lot to do with misinformation and miseducation of patients. Initially, antibiotics were seen as a cure-all for every single infection that humans ever encountered, like the common cold and acne. And they were prescribed as like a shotgun, and we prescribe them and use them to treat absolutely everything, even those things that would not be affected by antimicrobials, like viruses or other eukaryotic microorganisms that wouldn't be affected by an antibiotic. There are nearly 200 million prescriptions for antimicrobials written in the U.S. every year. That's a lot of drugs. And unfortunately, many individuals who receive those prescriptions don't follow the prescribed instructions for how to use those antimicrobics. Many people don't finish the prescription, which does not kill off all of the organisms. So now those organisms have been exposed to that antimicrobial at a lower dose and may develop a resistance or a tolerance to that level of antimicrobic. 75% of those 200 million prescriptions are for pharyngeal, sinus, lung, and upper respiratory infections that are caused by viruses. We know viruses are not affected by antibiotics. So we're exposing the normal organisms found in our body to these antibiotics. And over time, those organisms may develop a tolerance. They may acquire resistance to those drugs. And if opportunity arises in a situation where they are moved to a different location on the body or a different individual, they could cause an infection. So this shotgun approach is an additional contributor to this worldwide antibiotic resistance issue, and especially the use of broad spectrum antibiotics. We have prescribed broad spectrum antibiotics to treat everything because they have a wide range. However, there are organisms that were sensitive to more specific antimicrobics that could have been used, but since we have used these broad spectrum antibiotics so haphazardly, we have contributed to things like superinfections, where we wipe out the normal flora that help to protect us from pathogens, and this leads to other issues. It causes the development of resistance to our normal bystander microbes, those organisms that are naturally found there and are beneficial to us and help to prevent those pathogens from colonizing those locations. This has stopped in recent years, or at least has been reduced. More and more healthcare professionals are understanding the risk of antibiotic resistance and some of those practices that have contributed to that, and have become more aware of how they prescribe antibiotics. There is an additional problem outside of the United States. We export a lot of antimicrobial drugs, particularly to Latin America and Asia, and there are significantly reduced oversight there than there is here, and it's possible to purchase antibiotics over the counter and self-medicate and not follow a prescribed method of treatment. There are other issues as well. Antimicrobials are used readily in agriculture and in food products, and that exposure in our food also contributes to this developing problem with antimicrobial resistance. So with this problem, scientists have been looking at other ways to treat bacterial infections. There are a number of novel ideas that are being investigated. None of these are currently being used yet as marketable drugs, but it's important to be looking outside antibiotics for some of these therapies. So one potential treatment is targeting iron scavenging capabilities of bacteria. Another interesting idea is using RNA interference strategies. So there are regulatory RNA molecules that we know control or regulate gene expression, and those are currently being investigated as potential ways to control the expression of certain genes like resistance genes and other activities like metabolism within pathogens. There are also mimicking molecules called defense peptides, and these are produced by the host. These are produced by our immune cells, and scientists are looking at possibly manipulating these peptides or enhancing them during infections. Another interesting therapy is the use of bacteriophages to kill bacteria. And this practice has actually been used for a very long time. When Western medicine decided to use antibiotics, Eastern European countries took a different approach and started investigating the use of bacteriophages. And we learned in chapter 5 that bacteriophages are very specific to the bacterium that they target. And these countries, especially Poland and the Czech Republic, have been using bacteriophages very effectively to treat a number of infections like ear infections, sinus infections, upper respiratory infections, just a number of different infections using a bacteriophage cocktail, a variety of bacteriophages that target different aspects of a specific microbe and result in its death. So these are some pretty exciting ideas to help address this growing antibiotic resistance. There are a number of things that we can do to help our bodies while we are on an antimicrobial. Antibiotics can be harmful to our natural normal microbiota. And so there are a number of things that we can do to help enhance that and prevent pathogens from colonizing those empty places. And the first way is through a probiotic. So probiotics are live microorganisms that are fed to animals and humans to improve the intestinal biota, and they can help to replace microbes that are lost during antimicrobial therapy. They help to augment and support the biota that are already found there. Taking live organisms is very safe. These organisms are not pathogenic, but are normal organisms that we typically find in the human gut. And they're also useful to help manage food allergies. Another thing that we can do is the use of prebiotics. And prebiotics are not organisms, but they're nutrients. And they are nutrients that help to encourage the growth of the beneficial microbes that are already found there. And fiber is a great prebiotic because our normal intestinal cells cannot break down fiber, but our microbes can, and they do a great job of that. And these nutrients help to feed the normal flora that is already there and help to encourage its growth. Another thing that can help us naturally to fight some of these infections is fecal transplants. And this procedure is used to treat recurrent C. diff infections and ulcerative colitis. And what is involved is a transfer of fecal microbiota from a healthy patient via colonoscopy. And that fecal transplant introduces those healthy normal flora organisms to the infected patient to help fight the Clostridium difficile that is causing the infection in the intestine, and also to recolonize the intestine to prevent future infections. And work is currently underway to develop a pill that contains those species that would naturally recolonize the colon. So it would be less invasive. You wouldn't have to have a colonoscopy, but you could just take a pill to add those flora back to your intestine. And that is the end of section 10.3. These are our checking in questions, so make sure you go through these, double check that you can respond to them correctly. Also, make sure you look at our learning objectives for this mini lecture. And here's our critical thinking question. Does antibiotic use cause antibiotic resistance? Why or why not? So think about that question, refer back to the text, and then come back here once you've got a good answer and check your answer on slide 17. And hopefully this video was helpful.